Alan Maxam and Walter Gilbert proposed a sequencing method in 1977 based on chemicals that attack purines and pyrimidines. First, DNA is denatured with heat to obtain a single strand. DNA strands are labeled with phosphorus 32 on the 5' end. Different reagents break DNA by removing a base from its sugar through alkali or amine catalyzed beta elimination reactions. Chemical treatments take place in order to identify the sequence. Dimethyl sulfate methylates the 1' in DNA, whereas dimethyl sulfate with formic acid clips both 1' and adenine nucleosides. For pyrimidines, hydrazine reacts with thymine and cytosine, which are hydrolyzed and DNA is clipped. Moreover, with the addition of sodium chloride, thymine and hydrazine reaction is suppressed, obtaining clear cytosine bands. Piperidine is then added to every tube for its transition reaction. Finally, electrophoresis takes place and fragments migrate in an acrylamide gel. Through X-rays, bands show the location of radiolabeled DNA. DNA footprinting is a useful technique used for the analysis of DNA interactions determining the sequence of DNA binding proteins. Chemical reagents cleave to DNA at every base pair. By this method, a demonstration of the formation of weak or strong complexes between DNA polymerase and its accessory proteins can be observed. A DNA template with the added protein will run on the gel with a break in it, a footprint. Maxam-Gilbert DNA sequencing is run allowing the prediction of the exact location of the ligand binding site. It has also been used to identify binding sites of drugs and ligands bound to DNA molecules, especially the ones which don't leave a record of their residence position on DNA.
Viroids are a small, distinct class of infectious single-stranded non-coding RNA molecules. They're 50 to 80 times smaller than any virus, and they can cause diseases in various important plants. These molecules use self-cleavage structures that can cause disease via non-translatable RNA. The Maxam-Gilbert sequencing method has been used to better understand the processes involved in disease caused by viroids, as well as how they work and their adaptability. Potato spindle tuber disease can drastically reduce crop yield in crops such as potatoes, tomatoes, and eggplant. In potatoes, as much as 65% of an infected crop can be lost due to this disease. Theodore Diner and William Rammer were able to use the Maxam-Gilbert sequencing method using tomato plants to find the agent of this devastating disease, viroids. The specific viroid they discovered that was later named PSTVD, or the potato spindle tuber viroid. They were able to reveal the identity of said pathogen using a combination of density gradient centrifugation and enzymatic assays. This equipment that you can use to get results from this method is the Thermoscientific OWL dual gel vertical electrophoresis system, or the Fisher brand power supply Mini 300 volt plus.
The Sanger and Coulson method, also called dideoxy or enzymatic method. In this case, a copy of the DNA to be sequenced is made by the Klenlow fragment of DNA polymerase. The template for this reaction is a single-stranded DNA, and a primer must be used to provide the 3' terminus for DNA polymerase to begin synthesizing the copy. The production of nested fragments is achieved by the incorporation of a modified DNTP in each reaction. For DNTPs, one for each nitrogenous base, are included in a series of 4 reactions. The concentration of the dideoxy is such that it will be incorporated into the DNA chain infrequently. Each reaction produces fragments terminating at a specific nucleotide, and the 4 reactions provide a set of nested fragments. The DNA is labeled including a radioactive DNTP in the reaction. This enables more sequence to be read from a single gel than the phosphorus 32 labeled DNTPs.
Some applications for the Sanger method is an analysis of changes in the DNA methylation patterns associated with cancer and other pathologies. This method has been very important for the detection of 5-methylcytosine for more than 25 years. This allows the detection along the short genomic DNA region of up to 400 nucleotides. It's demonstrated the feasibility of the direct PCR sequencing workflow for the analysis of methylation in bisulfite converted DNA from cell lines and FFPE samples using reagents, equipment, and software. With this, we have faster results of methylation studies and opens possibilities for uses in retrospective research. Sanger sequencing is also founded in the validation of CRISPR-Cas9 genes. The CRISPR and the TALEN editing techniques largely allow specific genomic modifications, since the repair process is not completely accurate in genome editing and fragment analysis. Here, Sanger sequencing is used to determine the efficiency of CRISPR-mediated genome editing. This sequencing is very valuable as it is very accurate in simple and cost-effective workflow. In fragment analysis, it can detect insertions and deletions quickly.
Using Sanger sequencing, a research group in Germany investigated and confirmed the presence of SARS-CoV-2 RNA in wastewater in 9 different wastewater treatment plants around Germany in April 2020. Other studies have shown that coronavirus can be found in the feces of infected individuals, suggesting that wastewater could be a possible important source of infection if not treated properly. The researchers observed a greater presence of SARS-CoV-2 RNA in solid waste rather than liquid waste before wastewater was treated. A cell culture was used to see if there was still active and infectious forms of the virus in these waste samples, or if it was just RNA fragments. Based on their results, the group found that it could be possible that one could become infected with coronavirus through contact with untreated waste, though further research is required to confirm this, as well as to see how coronavirus affects public water supply.
The separation of the DNA fragments created in Sanger-Coulson method is achieved by PAGE, the polyacrylamide gel electrophoresis. Here we show the vertical electrophoresis system made by Thermo Scientific Fisher with its due power supply.
Today, the most widely used method for acid sequencing nucleases is the Sanger method. However, it is fair to say that the method of Maxam and Gilbert is the most suitable for determining the sequence in the short DNA fragments, because here you can determine the sequence from the first base. In contrast, Sanger method only allows reading to starting from 10 to 20 base,